we're very happy to have our speaker, who is Karen, who is currently at Stanford University. And I'm going to introduce her in a minute. But before that, let me just tell you a little bit about myself, but more importantly, Create and Learn. I know many of you have been taking classes with us. So my name is Jessie. I learned computer science in school, really loved it. And we started Create and Learn program a couple years ago and been teaching lots of students worldwide. This is our speaker, Karen. She is a researcher at Stanford University. She got her bachelor degree from UCLA in microbiology. So we actually went to the same school together. She has some very exciting thing coming up in the fall. I'm sure she'll be sharing with you guys as well. With that, Karen, would you like to get started?
Hi, everyone. Can you see me? Hi, I'm Karen. It is so nice to meet you. I'm so happy to be here with you guys today. So, as Jessie mentioned, I work at Stanford University. I'm a researcher in the laboratory of Dr. Howard Chang. And just like you guys, I'm actually also a student. So I'm actually going back to school to get my PhD in biomedical science. And today I have a presentation for you guys on genetics. And I'm sure a lot of you have probably heard of a molecule that we call DNA, but how many of you have heard of this molecule that we call RNA? Because today I'm going to tell you about RNA. Okay, I see a lot of you guys in the chat are saying that you know about DNA. I think that's awesome. So RNA is basically like the cousin of DNA. So it looks very similar to DNA, but it actually has a completely different job. And what I study in Howard's lab is actually RNA biology. So today you guys are going to learn to be molecular engineers and RNA biologists. And I'm also going to tell you a little bit about viruses, because I think a lot of you are probably having to stay home from school right now because of coronavirus, right? So we'll also talk about a little bit about the genetics of viruses and what is happening there. Okay, so let's get started. So first I want to tell you a little bit about the lab. So this is my lab. We're at Stanford University and we study the human genome. So basically we are focused on studying how your body maintains its DNA to keep you healthy and to keep your cell running correctly. So let's get started. Let's talk a little bit about what is genetics. So I mentioned this at the beginning of the presentation, but genetics has to do with DNA. So first I want to ask you guys, when you think of the word genetics, what do you think of? Can you tell me a little bit about what you picture when you think of genetics? So me personally, when I think of genetics, I think of Jurassic Park and like dinosaurs. DNA? Yeah, that's good. What makes you you? That's awesome. People, genes, helix. Oh my God, you guys are so smart. Okay, let's talk about genetics. So as biologists, when we talk about genetics, we think about genetics as the study of heredity. So basically, this is how characteristics are passed down from one living organism to another. So I know Jessie asked you guys earlier in the chat, like, do people say that you look like your parents or, you know, your grandma or your siblings? This is all because of your genetics. Okay. All right, guys, why do you think studying genetics is important? Can you write it in the chat? Yeah, coronavirus is definitely really important to genetics right now. Human ancestry, disease, being sick, genes, to know where you come from. Yeah, totally. Genetics is super important for all of those things. That's completely accurate. All right. So one big reason why people are interested in studying genetics, especially doctors and scientists, are that we are interested in learning how to diagnose, prevent, and treat certain diseases. For example, you know, you might have heard of Alzheimer's disease. Alzheimer's disease is actually a genetic disease. So there are certain genes which make you more likely to get a disease or not. So, and cloning, oh my gosh, that's awesome, totally. All right, so another reason why genetics is important is it also helps us with forensic science. So for example, if a burglar leaves his DNA behind at the crime scene, maybe if they leave their hair behind, your DNA is unique, so it can actually be traced back to you. So the police can actually use the DNA to help find the culprit of the crime. Let me see. So have you guys also heard of Ancestry.com? Because genetics is also really important in understanding human history. Yeah, a lot of you guys have heard of Ancestry.com. So Ancestry.com is basically a website that helps you find your long lost relatives. So your relatives who you might not know about or who live in other places can actually be found if you have similar DNA. And that's all because people in families tend to share very similar DNA. So you can actually understand the history of who you are and where you come from. Aw, thanks. Someone said they like the pictures in the slides. That's great. Aw. All right. So when we get started talking about genetics, I have to tell you guys about something called the central dogma. So what is a dogma anyway? So unfortunately, it doesn't have anything to do with actual dogs, even though dogs are really cute and awesome. The central dogma is actually defined as a set of rules or principles that we understand to be true. So for example, in astronomy, one of the rules of astronomy is that the Earth orbits around the sun. The sun doesn't orbit around the Earth. So that would be a rule of astronomy. Okay? So what is the central dogma in biology? So in biology, we think of the cell as being like a factory. So do you guys know what cells are? So cells are basically the most basic, simple units of life that exist. So organisms that are really complex, like humans, we have hundreds and thousands of cells that all make up our bodies, and our cells do different jobs within our bodies. And they all work to help us keep us healthy and strong. But really simple organisms like bacteria only have one cell. And the cell is like a factory. It's constantly reading our DNA to make things out of it. And the central dogma is like an assembly line. So when the cell reads our DNA, it puts that information into an assembly line that it uses to make things. It's like ice cream sandwiches in a factory. So the central dogma in biology works like this. So you start out with a molecule called DNA, which is basically all the instructions that you need to build life. DNA becomes mRNA through a process called transcription. mRNA becomes protein through a process called translation. You don't need to worry about the words transcription and translation right now, but I just want you guys to know that the concept is that DNA becomes mRNA, which becomes protein. So this is the process by which the information in genes is read by the cell. So the molecules DNA, RNA, and protein are central to life on Earth. DNA stores the instruction for building all living things like trees and flowers and cats and dogs. Proteins drive the chemical reactions for life. So until recently, RNA was just thought to be a simple messenger, and they thought that its only purpose was to carry instructions. So this guy, mRNA. But actually, mRNA is really cool because it can do a lot of different things. It can drive chemical reactions, just like proteins, but it can also store genetic information, just like DNA. So I personally think mRNA is really cool. I'm an RNA biologist, so I'm a little biased, but we're going to talk mostly about RNA today. All right, so let me ask you guys something else. So let's talk about heredity and how traits are inherited. So do you guys recognize this family in the picture? Where are they from? And what do they all have in common? Yeah, it's Ron Weasley from Harry Potter. How many of you guys have seen Harry Potter? Yeah? All right, cool. So one thing that all of these guys in these family, everyone has in common is red hair, right? So everybody in this family has the gene for red hair. So let me ask you guys, what do you guys think a gene is? Here's a hint, it's not a pair of pants. Okay, someone said the gene is a code, it's information, it's passed down from your parents, gene, genetics, info. Yeah, that's really good, it's DNA. Okay, so in genetics, a gene is defined as a small section of DNA that contains instructions for making our individual characteristics, like our eye color or our hair color. So when you inherit a gene, they actually come in different forms, which we as scientists called alleles. humans tend to inherit alleles in pairs. So we get one allele from our mom and one allele from our dad. So how many of you guys have a different hair color or eye color from your brother and sister? Can you say yes? Yeah? Yeah? Okay, a lot of people. All right. Yeah. I guess if you're an only child, it doesn't count. But maybe you have a different eye color from your mom and dad. Yeah, so if you have a different eye color from your relative, this is because you and your relative have different alleles. So if your brother has blue eyes and you have brown eyes, that means that your brother has the allele or the gene for blue eyes, but you got the allele or gene for brown eyes from your parents. So now that we know that a gene is a small piece of DNA with the instructions to make a protein, let's look at how this works. So let's say you have a gene for flower color. You have this DNA sequence as a set of instructions that tells the flower how to make a color protein. So this DNA sequence gets read by the cell, and the cell makes a protein that helps produce flower color pigments. These flower color pigments, when they're translated into the actual flower, make the flower look purple. So basically, when our cells read our DNA, they're learning how to make things that help us survive or give us our unique traits. DNA is where all your genetic information is stored. It's the instructions for life. So let's watch a short video about how your cell works as a factory, as an assembly line to produce all the molecules that are necessary for life.
If you could step inside one of your cells, you'd see something that looks a lot like a factory, building thousands of different molecular machines. It would have tiny assembly lines starting in the cell's nucleus and stretching out to structures called ribosomes. These assembly lines start with DNA and build RNAs and proteins. In a very real sense, you. DNA has all the blueprints for everything the cell will ever need to build. RNAs are assorted molecular machines that do all kinds of work in the cell. They carry components from place to place, ratchet up the speed of operations, switch other machines on and off, and relay coded instructions from the nucleus out to the factory floor. The other product, proteins, are strong molecular machines that do things like hold the cell together and send signals to other cells. There are two parts to each assembly line. The first builds RNAs and the second builds proteins. The process that builds RNAs is called transcription. It happens in the cell's central office, the nucleus, where DNA blueprints are stored. Let's say that a cell needs to make a transfer RNA, or tRNA, a triangle-shaped RNA which transports a building material called an amino acid. It starts at a tiny, specific region of DNA with the code that makes the tRNA we need. This region is called a gene. A protein machine inside the nucleus pries apart the weak bonds that hold the two strands of DNA together. RNA building blocks swarm in and form a conga line complementary to the DNA. This RNA strand now needs to fold up. In the world of tiny cellular machines, shape determines function. RNA's 4 bases bond to each other and give the tRNA its 3D shape. And voila, our transfer RNA is ready for action. It floats out of the nucleus and picks up an amino acid. We'll come back to our tRNA in just a moment. Now let's look at the second half of the factory's assembly line, making proteins from RNA, a process called translation. Remember the scrape? This cell needs to make thrombin, a protein machine which helps blood clot and make a scab. Thrombin is just one of the tens of thousands of proteins your cells can make. Before translation begins, the cell transcribes a special type of RNA called mRNA, or messenger RNA. This mRNA carries the code for thrombin out of the nucleus and onto the cellular factory floor. Our mRNA drifts until it runs into a ribosome, a protein-making machine. The ribosome clamps down around the mRNA. tRNAs, just like the one we made earlier, drift in. The messenger RNA carries a coded message, which the tRNAs translate into amino acids, the language of proteins. This process, translation, proceeds down the mRNA, creating a chain of bonded amino acids. Just as with RNA, molecular shape determines molecular function, so these amino acids fold up into the protein's 3-dimensional form. Finally, the ribosome comes to a bit of code that says, okay, we're done here, and releases its completed protein into the cell. So there you have it. DNA encodes genes to make RNA machines, and the ribosome translates special mRNA, which in turn makes protein machines. Unlike most factories, your cell uses just 2 processes to make tens of thousands of different molecular machines. It's like one factory that can build toasters, phones, cars, and even repair itself.
All right, so let's move on. So they said something that was really important in the video, which is basically that a molecular shape is linked to its function or its job in the cell. So that's a really important concept. I want you guys to really, really remember that shape is linked to a molecule's job. Okay, so now let's look at a strand of DNA, which is the first step in our central dogma, our #1 rule. So our genes, our instructions for making things, are arranged in given positions along our DNA. So here we have the part of your DNA that is the gene for height, and here's the part of your DNA that is the gene for eye color. So DNA is our genetic blueprints, and every single cell in your body has the same DNA. Your DNA is unique to you. So the other thing that's really important about DNA is its shape. Do you guys notice that it's in your double helix shape? So a lot of people recognize DNA by its shape because there are 2 strands wound around each other. And this double helix is actually really important to DNA's job because DNA is meant to store information, but to do it in a way that the information is protected really well and isn't lost or damaged so that the cell knows what it needs to do in order to survive. So when DNA is wound around itself in this double helix, it actually makes it really stable. It's really hard to lose the information in DNA when it's in this shape. Okay, so DNA has its own language. DNA has a code. So this code is written in the language of 4 bases, which are paired together. And the rule of DNA is that certain bases can only pair to each other. This is a really important rule. It's called base pairing. So this yellow base, adenine, can only join to the orange base, thymine. The red base, cytosine, can only join to the blue base, guanine. All right? So the other thing that I want to tell you guys about DNA is about its structure. So these bases are held together by a sticky glue called a hydrogen bond. And so the bases themselves are made of nitrogen. The backbone of DNA, this green part, is made of sugar and phosphate. So that's this red circle and this blue pentagon. This is the sugar phosphate backbone of DNA. But when you think of DNA, you think of it as a code or a sequence, right? So when your cell reads your DNA, it's a lot like when you read a book in real life. So when you read a book, what you're reading are sentences and words on a page, right? So in DNA, the individual order that the bases are placed in, these pairs, these are like the letters in a book. When you put the bases together, you get a gene. A gene is like a word in a book. When you put all the letters together in a certain order, they start making words which send a message. It's the same with bases and genes. So when you put the bases together in the form of a gene, it sends a message to the cell. Okay, so let's play a game with you guys. All right, so we're gonna play a game to see if you can crack the genetic code. So here are the rules for pairing bases on the left, and I'm gonna ask you guys if you can play this game with me, okay? Yeah, you're gonna combine the pairs. Okay, so if I have the base adenine, what pairs with the yellow adenine? Can you guys type it in? Okay, all right, awesome. If you guys typed thymine, T, into the chat box, you're correct. Wow, good job. You guys are really smart. Okay, cool. All right, let's try another one. What if I give you a red C for cytosine? What does cytosine pair with? That's right. Cytosine pairs with guanine, the blue base. All right, let's try one more. Okay, what about orange thymine? What does thymine pair with? Right, thymine pairs with adenine. Okay, you guys are awesome. You were really smart. I think that problem was too easy. Let me give you a harder one. See if you can get this one. Yeah, oh my gosh. Okay, this is so cool. You guys are molecular detectives already. All right, so if you typed CCAT into the chat box, you guys are correct. Awesome, you guys are awesome. Cool. So DNA is stored in the nucleus, right? So where is DNA stored? And the nucleus is basically like the headquarters of the cell. So remember when I said that DNA stores all your genetic information and it has to be protected really well? The cell does this by storing your DNA in the nucleus. So basically, when the DNA is in the nucleus, it's a lot harder for the DNA to be damaged or lost from the cell. It's a way for the cell to keep your DNA safe so that it doesn't lose any of the instructions it needs to survive. Okay, here's another question. How much DNA do you have? A lot. So the answer is, if you actually uncoiled all the DNA and all the cells in your body, it would stretch over 10 billion miles, or to the sun and back 61 times. So you have a lot of DNA, and a cell is all really, really small, and the DNA has to fit into the nucleus, which is even smaller than the cell is. So how does the DNA fit into the nucleus if it's like really long? So the answer is that DNA is supercoiled around a protein called a histone so that it takes up less space inside the cell. Supercoiling is basically when the DNA is wound really, really, really tightly so that it takes up less space. So if you guys have played with a yo-yo before, supercoiling is a lot like a yo-yo. So let's imagine that the yo-yo string is DNA, and then the body of the yo-yo is like the histone protein. So even though the yo-yo string is really long when you throw the yo-yo, when you wrap it up along the yo-yo body, it becomes a lot shorter. It's really small and it takes up less space. All right, so let's move on. So I know a lot of you guys have been asking about when are we going to get to RNA. So RNA is the next step in the central dogma. So let's talk about RNA now. Okay, so RNA is read from DNA in a process called transcription. So this is showing the steps in the central dogma, basically, but an enzyme called RNA polymerase runs along the strand of DNA to make something called an mRNA, and then it falls off and releases the mRNA. All right, let's talk about RNA, my favorite molecule. So transcription is the process by which DNA is made into RNA. After transcription, mRNA leaves the nucleus to be translated. So did you guys notice that there's an M in front of mRNA? That stands for messenger. This mRNA is a messenger. It's like the cell sending an email from the nucleus with instructions to the rest of the cell for what to do. Okay, so let's look at RNA compared to DNA. So I'm a little biased. I think RNA is really cool. Okay, so what are some of the differences that from RNA and DNA that you guys see in this picture? Yeah, I know the words are really complicated. I'm sorry, guys. I didn't choose these names. Yeah, one strands. Ribonucleic acid. So there's no blue. Yeah, deoxy. Yeah, totally. Okay, these are awesome answers, guys. You're doing really well. Okay, so the biggest difference I think people saw in this picture is that DNA is double-stranded, while RNA is single-stranded. But you can see that they look kind of similar, right? They still have those nucleotide bases running along the strands. But there's one really big difference between DNA and RNA. And that is, although they have the nucleotides, instead of thymine, RNA has a uracil, this orange base right here. And so basically, DNA and RNA, when they base pair, they have really similar rules. So in DNA, adenine pairs with thymine, but in RNA, adenine pairs with uracil. But DNA and RNA all share the bases adenine, cytosine, and guanine. The other big difference is that DNA has the sugar deoxyribose, which is where the D in DNA comes from. RNA has the sugar ribose, which is why it starts with an R instead of a D. Okay, so once again, I think RNA is a lot cooler than DNA. And that's because I think RNA is cool because it can make a lot of different shapes. Remember when we talked about how molecular shape was related to a molecule's job? RNA is single-stranded, although sometimes in viruses it can actually be double-stranded. And because RNA is single-stranded, it gives it the ability to make a lot of different shapes, way more than DNA. So DNA has the double helix structure, which makes it really stable. But because RNA is single-stranded and can also base pair, it can fold in on itself, like here in this picture, and it can actually bind with itself. This is called RNA folding. And RNA and DNA, their stability is a lot like the differences between a Twizzler and a fruit by the foot, if you guys have eaten those candies. So DNA is like a Twizzler. It has a bunch of different licorice strands that are wrapped around itself. Let's imagine RNA is the fruit by the foot, and it's just one strand of candy, right? So when you try to bend a Twizzler, it's a lot harder than bending a piece of a fruit by the foot. Even though a fruit by the foot is kind of sticky, it can stick to itself. And that's because the DNA is all twisted around itself. So it's a lot harder to mess with the genetic information in DNA. So once again, instead of a thymine like DNA, RNA contains the base uracil. RNA uses the sugar ribose instead of deoxyribose like DNA. And also, RNA can leave the nucleus to make proteins, but DNA is kind of lazy and it sticks around the nucleus to be protected and safe from degradation. Ah, someone said I sound like the Amoeba Sisters. I love the Amoeba Sisters. All right, so does anybody have any questions? Let's check the Q&A box. What does tRNA do again? tRNA helps in making proteins. So DNA turns into RNA, which turns into a protein. Oh, someone asked, can people lose their genetic info even if the DNA is still in the double helix form? Yes, DNA can actually still be damaged even when it's in the double helix, and that's actually part of what my lab studies is. How do we fix your DNA when it gets damaged? You know, like what is the effect of that on the cell, things like that. Okay, let's move on. Okay, so we talked about RNA taking on a lot of different shapes because they can take on a lot of different shapes. They can take on many different jobs in the cell. So for example, we have mRNA, which we talked about earlier. This is like the email that the cell sends from the nucleus that gets produced after the cell reads the book of its DNA. You have ribosomal RNA, which is in the ribosome and helps make protein. And then you have tRNA, this guy. He looks like a cross and he helps transfer amino acids, which are the building blocks of proteins. Okay, so I want to talk about a type of RNA called a riboswitch. So because RNA takes on a lot of different shapes, scientists are trying to exploit this to treat different diseases like cancer. So here on the left, this is actually a piece of real RNA. This is a structure that was predicted by a computer program called Vienna. So do you guys notice that this RNA is like really complicated and kind of looks like a key? It has a lot of different loops. It has a long stem. Sometimes it even has loops inside the loops. It's really complicated compared to DNA, which is just a double helix, right? So this is something that I find really cool about RNA, is the fact that it can take this complicated shape. And so in RNA, the different base pairs actually have bonds of different strengths. And this is actually something that's really important because some of RNA's bonds are weak and they can easily break and allow the RNA to transform. So RNA is really unstable, unlike DNA and protein, which is all really stable. So RNA, because it can transform, what scientists do with a riboswitch is they engineer a type of RNA that can recognize some sort of molecule. Yeah, so in cancer, potentially, if there's a molecule that's unique to cancer, maybe the RNA, the riboswitch, can bind to that molecule and recognize it. When the RNA recognizes the molecule, it changes its shape to shut down the cancer cell. Okay, it looks like some people are getting a little confused, but for now, the most important thing to remember is that RNA can take on a lot of different shapes and fold in on itself. Yeah. Okay, let's talk about RNA and viruses. This is really important, okay, guys? So when a virus encounters a cell, this is a virus, the virus actually injects its genetic material inside the cell. The virus is basically just a little packet of genetic material, which can be DNA or RNA, inside a protein shell. So here you can see the virus is merging with the cell membrane, and then its genetic material is going inside the cell, right? Yeah, so the cell turns into a virus duplicator, someone said. Oh, wow, you know what a bacteriophage is. You're really smart. Okay, cool. So what happens when the genetic material of the virus enters the cell is that the cell starts to think that the virus's genetic material is its own. It's not smart enough to tell the difference. So instead of making all the things that the cell needs to survive, the cell gets tricked and starts making stuff for the virus instead. So this is what happens with coronavirus too. So instead of making all the things that the cell needs to survive, the cell starts focusing on making things for the virus. And then it gets really sick because it's not making things for itself anymore. Yeah. And so actually, viruses have been around for a really, really long time. You actually have like a ton of viruses around every day. And our cell is really smart, and it evolved all these ways to recognize the viral material and keep itself safe by cutting up the virus's genetic material. So the problem is when you encounter an RNA virus, so one example of an RNA virus is the coronavirus that's making everybody sick. Another example of the RNA virus is the flu. So you guys know how you have to get a flu shot every year? It's because compared to DNA, RNA is really unstable. That means it can change its genetic sequence really easily. It can evolve and mutate, which means change its sequence, really fast. And so every year the flu virus changes its genetic sequence. That's why you have to keep going back and getting a flu shot, right? The thing about RNA viruses is because they also evolve so fast, they evolve so fast that they can jump all the way from animals into humans. And so people think that's what happened with coronavirus, is that it came from an animal into a human. And because it came from an animal into a human, humans weren't able to build up the defense system or the immunity to fight the virus. So a lot of people started getting infected and started getting really sick because we just haven't had time to evolve immunity against this new virus, right? It's completely new to our bodies. We've never seen it before. So this is a cartoon of coronavirus, right? So this new coronavirus is an RNA virus. Now we know what RNA is. And I told you guys that RNA makes many different shapes, right? And there's a lot of difference or there's a lot of interest in making an RNA-based vaccine against the virus. So this is actually something that you guys can help with. So even though we're kids, maybe if we can help solve the structure of the virus, we can help scientists develop a vaccine to fight the coronavirus. And so basically what scientists think is that if we can solve the structure of the RNA in coronavirus, we can make a vaccine made of RNA that looks like coronavirus, but doesn't make you sick. And then if you show this RNA to the cell will generate kind of a picture. It'll be able to recognize coronavirus when it actually infects the cell and defend itself, because it will know what coronavirus looks like. So this is the way that vaccines work. And so if we can give the cell that picture, if we can teach it to recognize coronavirus, maybe we can defend ourselves and keep them safe from getting infected. Okay, so my lab is helping develop this game called Eterna, which helps fight against coronavirus by predicting the structure of the vaccine, of the RNA. So let me see if I can show you guys a video about this.
A major scientific question is to be able to predict what shape an RNA will fold into in practice. And we know that our models aren't good enough to do this. So we have computer models, but they don't explain how RNAs fold in practice. And what a scientist would really do is perform experiments and see how those experiments work and then repeat and so on and so forth. And what Eterna does is open up this whole scientific cycle to the public and allow them to propose experiments, see the results, and then propose more experiments. And ultimately, we hope, help us generate hypotheses about when RNAs fold the way they do and why they do.
Okay, so we don't need to watch the whole video, but what you guys basically need to know is that we are trying to predict the structure of this RNA virus through a video game that we call Eterna, which was developed by Riju Das here on the left at Stanford University. Okay, so let's watch another video that brings all this info together.
A tiny molecule packed with a 4-billion-year-old code may hold the answers to some of biology's biggest questions. This molecule is called RNA, and scientists are just beginning to understand all the amazing things it can do. Better yet, there's a way that you can help solve the mystery of how RNA works, just by playing a game. You may be familiar with RNA's more famous cousin, DNA. DNA is a double-stranded helix made of 4 building blocks called nucleotides. These nucleotides code for all of the genes that make fish, fish, trees, trees, and you, you. RNA is also made of nucleotide building blocks, but it's a single-stranded molecule. Scientists were initially fooled by RNA's simplicity. They thought that it was little more than a messenger between DNA and proteins. The truth is far more interesting. RNAs are versatile cellular machines that can do what both DNA and proteins do, and more. They can switch genes on and off, defend cells against attackers, alter other biomolecules, and store genetic information. Every year, new RNAs are discovered, and the list of their Swiss Army knife-like skills expands. RNAs function by folding up into shapes. Some shapes bind to DNA, some cut up RNAs, and still others bond pieces of protein together. In each case, an RNA's shape determines its function in a cell. If we could predict exactly what shape any RNA sequence will fold into, we could harness RNA's power. For example, we might design an RNA that turns off cancer-causing genes. Scientists have tried to use computers to solve this problem, but they haven't had much luck due to the sheer number of possible combinations. And that's where you come in. You can do something that computers cannot: find the patterns that rule RNA design. To take advantage of these distinctly human skills, computer scientists and biochemists teamed up to create a game called Eterna that allows anyone, not just experts, to find these patterns. The challenge of the game is to design RNAs that fold into specific shapes. The creators are counting on gamers, Sudoku solvers, artists, and other creative individuals like you to bring fresh perspectives and skills needed to crack the RNA code. You'll start off in Nova's RNA lab, playing as a molecular engineer who must solve RNA puzzles to complete a series of challenges. Once you learn the basics there, you can join Eterna, where you'll encounter even more challenging puzzles. If you prove yourself to be an expert player by earning 10,000 points, you can join a community of elite RNA engineers who craft real molecules designed to help solve RNA's many mysteries. Each month, thousands of these designs are synthesized and analyzed in a lab in California. If your design is chosen, you'll receive a score and data that will tell you how well your RNA performed. Solving the problem of RNA folding can change the world. One of your designs could help create an RNA that destroys the HIV virus, or a microscopic computer made of many RNAs that detects and defeats cancer before it spreads, or a self-replicating RNA that shows how life began, or maybe even a breakthrough that no one has ever dreamed of. So what are you waiting for? This RNA is not going to fold itself.
Okay, so now you guys know why RNA is important and why we're interested in solving the structure of this virus, right? So are you guys ready to play a game? Now we get to be molecular engineers and we get to help the scientists in the lab to solve the structure of this virus. Okay, let's do it. So I'm gonna put this link in the chat, but first I wanna walk you guys through this tutorial. When you click in this link, it's gonna open up this tab, right? Yeah, it's a science party. This is awesome. Okay, so when you launch the game, you're gonna go into the virtual lab, and what's really cool is that if your design in Eterna is good enough, then they'll actually make your molecule in real life and test it in a Stanford lab. So as an RNA engineer, this is what we're gonna do. Welcome to the virtual lab bench. So it's basically gonna have all these tooltips that will walk you through how to fold RNAs in the lab. So this is a strand of RNA. These are the bases that we learned about: adenine, guanine, uracil, cytosine. And then we're going to use this palette to create mutations and paint the RNA strand, right? So you click on the red in the palette and then follow the instructions in the game to make it. Okay, cool. All right. You guys want to try this for yourself? It will give you all the tools you need and the basic biology that you need to play the game. So when you guys play from the virtual lab bench, eventually you can move up to Eterna to start solving harder and harder puzzles. And there's a community of players in Eterna who spend a lot of time talking about how to invent new molecules to be medicine for different diseases like tuberculosis and COVID-19.
Eterna is a puzzle-solving game that reveals the mysteries of one of the most powerful molecules in biology, RNA. RNA is the companion of DNA, taking genetic information and putting it to work inside our cells by folding into complex structures. Smart RNA has the potential to transform medicine, tackling some of the most prevalent and deadly diseases on the planet, like tuberculosis, malaria, and cancer. But scientists don't fully understand its behavior. That's where Eterna comes in. Players use the 4 nucleotides that make up RNA to design a pattern that forms into a target shape. As they progress, the puzzles become more complex, and the challenges reflect the mechanics of nature. After gaining experience, players reach the lab, where their designs are manufactured and tested at Stanford University, often tens of thousands at a time. The experimental results are then returned to the players, so they can improve their designs and become scientific collaborators, joining professional scientists on the frontiers of biochemistry.
All right, citizen scientists. So are you ready to help out with folding these different RNAs? Okay.
Vaccines are a way of exposing your body to an inactive form of a pathogen or a weakened form of a pathogen. Since it's inactive or weakened, it prevents you from actually developing the disease from the vaccination itself. However, your body still launches an immune response. By launching an immune response, which includes the production of antibodies against the pathogen, the immune system will retain memory against the pathogen. That way, if your body ever encounters the real active pathogen, your body will be familiar with it already, and therefore we say that your body already has immunity against the pathogen. This can result in allowing your body to launch an attack against the real thing more quickly and efficiently. We take for granted the amazing work of vaccines in keeping people from developing devastating bacterial and viral diseases. Thanks to vaccines, some horrible bacterial and viral diseases are considered eradicated in some areas, but it is important for them to be continued to prevent some of these diseases from reemerging. There's also another really important thing that you should know about vaccines. Some people may not be able to receive certain vaccinations. From someone whose immune system is severely compromised because perhaps they have an illness of some type, to a newborn baby not yet old enough to receive many vaccines, to a pregnant mother. These vulnerable populations rely on something known as herd immunity. That is, if others around them are vaccinated against a certain pathogen, that may offer them a degree of protection against that pathogen because it cannot be easily spread to them. So vaccinations not only offer protection for the person receiving them, but also they can offer protection for others around them.
Okay, cool. So if you guys are interested in playing the game after this, I'm going to include some links to our virtual labs and to Eterna and the COVID-19 OpenVaccine Challenge. If you guys think that RNA is really cool, then you can also learn more about RNA at Khan Academy, where there's like a series of lessons about viruses and their history and all the things that you can do with RNA.
So maybe whoever have a question, if you want to raise your hand in the chat, or raise your hand, then Karen can call you. Do you see them raising hand, Karen?
Let's see. Let Dia ask her question. Hi, Dia. Can you hear us okay?
Yeah, I can hear you.
Okay, what's your question, Dia?
So I just wanted to know, like, can you inherit genes from like siblings, or can you only like inherit from parents?
Okay, that is a great question, Dia. And the answer is that you can only inherit genes from your parents. So your DNA comes from your parents, not from your siblings. Okay, so that was a great question. Let's ask another one. How about Kundana?
Hello?
Hi, is this Kundana?
Yeah.
Hi, Kundana. What was your question?
Where's the RNA located and the DNA? Is it all in your body?
That is actually a really great question. And that's actually part of the project that I'm researching right now. So remember how I said that DNA has to stay in the nucleus because it's protected there? RNA can actually travel to all different parts of the cell. So there's some RNA that actually stays in your nucleus and has jobs inside the nucleus, but there's also RNA that has a lot of different jobs around the other areas of the cell. So the area outside the nucleus in the cell is what we call the cytoplasm, and there's tons and tons of RNA that are floating around in there. All right, how about Naman? Can you hear us, Naman? What was your question?
My question was, how did the central dogma get its name?
How did the central dogma get its name? That's a good question. I'm actually really not sure myself. Scientists decided to call it that because a dogma is a scientific word. It's like a terminology for a rule or a principle that people understand to be true within a field. But as for why people call it that, I think it's just scientific tradition. All right, good question, Naman. Hi, Josiah. Okay, can you hear us okay? Josiah?
Yes.
Hi, what's your question?
So my question is, can DNA or mRNA or RNA in the body be damaged?
Yes. So that's actually something that a lot of scientists, including myself, are really interested in, is it's really bad for your body when your DNA or your RNA gets damaged. With RNA, it's not so bad because RNA is supposed to be temporary. So an RNA is like an IM or like an email from the body. It's pretty temporary and it just transmits a bit of information. But when your DNA is damaged, then your cells lose control of all the things that they need to do to survive. And that can result in diseases like cancer, which come out of your DNA being damaged and your cell being unable to control its proliferation. And it makes people really sick. That was a really good question. Is this Saketh? Hi, Saketh. Can you hear us? Oh,
Yeah.
Hello. Hi. What's your question?
Oh, so my question was, so how long can like information on DNA be passed on?
That's a really good question. And this is going to be a really advanced answer. I know that I've given you guys a lot of information today, and it might be kind of a lot to take in. But basically, when you get older, during the process of aging, your DNA starts to get shorter and shorter and shorter, and you start to lose ends and pieces of DNA. And so this is really bad because when you're aging, as you use your DNA more and more, as you make more copies of it and it becomes shorter and shorter, you start to lose really critical information at the ends. So DNA is around as long as you have a cell, but it changes over time. And that's why people grow and age and all of these processes. Hi, Jessa.
Hi. So I know that viruses and bacteria are both like kind of bad, but which one holds like a stronger power over your body?
Which one holds stronger power over your body? Okay, that's a really good question. So, okay, so I guess this question will also be kind of complicated to answer. And also, I'm not a doctor, but it kind of depends on the virus and the bacteria. There are some bacteria which are really, really bad. Like, for example, tuberculosis is caused by a bacteria, and viruses are also really, really bad. But I'm sure sometimes you've gone to the doctor and you're really sick, maybe you have a cold or something, and you go to the doctor and they tell you you have the flu. And they tell you that because you have the flu, there's nothing that they can really give you because antibiotics are supposed to work on bacteria. They don't work on viruses. It really depends on the disease, but sometimes the doctor can't give you something to treat viruses because they work differently from bacteria. Vaccines, like the ones that you get when you're a little kid against things like polio or measles, are designed to help your body fight viruses when they see them. But in terms of which one is worse, it really depends on the disease. Dimitri, can you hear us?
Oh, hello?
Hi.
Hi.
Yeah, hi, Dimitri. We wanted to ask what your question was.
Yeah, so I know how, like viruses mutate. So is there like a certain percent chance like for it to mutate, or it just occasionally happens?
So every mutation is kind of random. That's a really good question. And a lot of people are asking the same question as you right now, is whether we can predict the mutation rate of a virus. We can actually playing Eterna, the game, will actually help scientists do that.
Okay.
The short answer is if something is a DNA virus, it's less likely to mutate than an RNA virus. And that goes back to what I was telling you guys earlier, which is that DNA is double-stranded and RNA is single-stranded.
Yeah.
Because of this strandedness, RNA is more unstable and it mutates a lot more quickly because it's less stable than DNA. So RNA viruses mutate faster than DNA viruses in general. As to whether I can give you a percentage, I can't really give you a percentage. I think that also varies from virus to virus, and the mutation chance is random.
All right, good to know.
Mm-hmm. All right, great question.
Thank you.
Mm-hmm.
So today we've been doing these, what we call the open classes, every single week. There will be more of them coming up. And many of you probably already tried out our free introduction classes. We offer a bunch of coding class, and you should go check it out if you haven't done it. This is our website at create-learn.us. And you can have your parents who sign you up for the open classes I mentioned earlier. And then we also offer online camps. All right. with that, I want to thank Karen for joining us today. That was awesome. I learned a whole bunch of things as well, and very, very interesting. And I do encourage everyone to go back and play the game. You know, you're going to help the scientists to do really, really interesting research, and you can learn a whole bunch of very interesting things yourself as well. Okay, well, thank you. Hope to see you guys again in future sessions.
Thank you, everyone. It was nice meeting you. That was awesome.
Bye.